Hello everyone! Today we're going to talk about the pentose phosphate pathway. So these are the pathways we have been talking about in class for the past week, and today's subject is the pentose phosphate pathway. So in the Where Are We section of our anabolic pathways, the pentose phosphate pathway is a pathway in which glucose is converted to ribose and deoxyribose for use in nucleotide biosynthesis. And what's not shown here is that the pathway also produces NADPH for use in fatty acid biosynthesis as well as other compounds. So what you should do at this point is make a list for yourself, maybe draw a diagram of all of the pathways in which glucose 6-phosphate is connected, either to a reactant or a product. So you should pause the video here to draw your diagram. Well, hopefully you came up with something that looks like this. I think I will illustrate this using this animated figure that we have here. So we saw earlier that when glycogen is broken down, we break it down to glucose 1-phosphate and it's isomerized to glucose 6-phosphate. Glucose 6-phosphate is converted to glucose 1-phosphate and is used in glycogen synthesis. Glucose is converted to glucose 6-phosphate using the hexokinase reaction. In gluconeogenesis, we take pyruvate and other 3-carbon compounds and convert them to glucose 6-phosphate in gluconeogenesis. In the pentose phosphate pathway, as we will see in a minute, glucose 6-phosphate is converted to ribose 5-phosphate. Glucose 6-phosphate is converted to pyruvate, oxidized to pyruvate in glycolysis. And glucose 6-phosphate can be converted to glucose in some tissues, notably the liver and the kidney, which express the glucose 6-phosphatase enzyme that's required to accomplish this transformation. And later we will learn that pyruvate will be further oxidized to acetyl-CoA and then will enter the citric acid cycle. And that's a subject for chapter 14. In chapter 17, we will look at how pyruvate is converted to amino acids. And we've already seen that pyruvate is converted to lactate. So in much the same way, pyruvate is at the crossroads as well as glucose 6-phosphate of metabolism. Okay, so let's go back to our PowerPoint. So if we think about pathways of carbohydrate metabolism, when we talked about glycolysis, we catabolize glucose to yield ATP. And we refer to ATP as cellular energy currency because we can use ATP to power endergonic reactions. But we can also think of NADPH as cellular energy currency because NADPH holds protons and electrons, or reducing power, and there are lots of reductive reactions that require the donation of protons and electrons. So the NADPH can be produced in the pentose phosphate pathway to provide the reducing power that's needed for those anabolic pathways. So what this means is that the cell has to regulate the balance between these two pathways. Sometimes we need more ATP, sometimes we need more NADH. And since glucose is the source of both of those things, the cell has to be able to balance what it needs at any particular moment. So this is a diagram of the pentose phosphate pathway. We will return to this at the end of the lecture because I think looking at it in this way is maybe not particularly enlightening. I think it will make sense at the end, so I'm going to come back to it at the end. It's a nice summary once we sort of understand how all of these metabolites fit together. But for now, I will say that the PPP produces ribose. We need ribose for DNA, RNA, but also coenzymes like NADH. FAD have ribose as part of their structures, and it will also produce NADPH for fatty acid and cholesterol biosynthesis. NADPH is also needed for glutathione and to reduce reactive oxygen species, especially in red blood cells. There are actually quite a lot of pathways involving NADPH. So fatty acid biosynthesis, cholesterol biosynthesis, neurotransmitter, and nucleotide biosynthesis, all of them require NADPH. And because NADPH can donate electrons, we can reduce oxidized glutathione and the cytochrome P450 monooxygenases. So NADPH is a really important metabolite for all of these pathways. And we can get it from the pentose phosphate pathway. We should note that NADH and NADPH are not interchangeable. Chemically, they look very similar. There's just a phosphate group instead of a hydrogen. But remember, NADH is produced in glycolysis. It holds temporarily the electrons and protons that are stripped from glucose. And those protons and electrons will eventually be donated to oxygen to form water in the electron transport chain. NADPH, by contrast, is the reducing agent used as a reactant for fatty acid and cholesterol biosynthesis. So very similar chemically, but metabolically they're very different. So we can divide the pentose phosphate pathway into two branches, the oxidative branch or stage 1. Here the goal is to produce NADPH, and we need to do this by oxidizing glucose 6-phosphate. And then we will look at the non-oxidative branch or stages 2 and 3. So let's look at the oxidative branch first. The first part of the pentose phosphate pathway, again, the oxidative branch or stage 1, is glucose 6-phosphate is oxidized. So if you look at this carbon right here, carbon number 1, we're going to take protons and electrons, donate them to NADP plus to form NADPH and H plus. And now we have an oxidized carbon. See here we have one bond to oxygen and one bond to hydrogen. Here we have two bonds to oxygen. So we form this lactone, which is oxidized. A dehydrogenase enzyme, remember, catalyzes redox reactions. So this is a redox reaction in which the glucose 6-phosphate is oxidized and the NADP plus is reduced. The second reaction in the oxidative branch, the lactone, will just do a simple hydrolysis using a gluconolactonase to open this up to the straight chain form. And here we can definitely see the oxidation. Remember here for glucose, if we open the ring, we get an aldehyde. And here it's a carboxylic acid. So this has clearly been oxidized. And the third step, we're going to use another dehydrogenase enzyme, and we're going to lose this carboxylate group as a CO2. And protons and electrons will be lost here, but they will be donated to another NADP plus to form NADPH. And we end up with ribulose 5-phosphate. So the products of the oxidative branch of the PPP are simply ribulose 5-phosphate and 2-NADPH. So for every one glucose 6-phosphate that enters the oxidative branch, which is only 3 reactions, one carbon from glucose 6-phosphate is completely oxidized to CO2, and the protons and electrons that are lost are donated to NADP plus to form NADPH. And we end up with a 5-carbon sugar, ribulose 5-phosphate. So 2-NADPH are obtained when one glucose 6-phosphate passes through stage 1 one time. Okay, so let's go on to the non-oxidative branch, or stages 2 and 3. Well, what are we going to do with this ribulose 5-phosphate? We're going to dispose of it in stages 2. In stage 3, we are going to convert the intermediates into compounds that can feed into either glycolysis or gluconeogenesis. So imagine we've gone through stage 1 3 times, and now we have 3 ribulose 5-phosphates. And what we're going to do is we're going to take 2 of them, and an epimerase is going to act on them. And then we're going to take the third one, and we're going to use an isomerase to convert the ribulose 5-phosphate to ribulose 5-phosphate. So we're going to go through stage 1 3 times, get 3 ribulose 5-phosphates so that we end up with 2 xylulose 5-phosphates and 1 ribose 5-phosphate. So let's look at these reactions in more detail. So here's the ribulose 5-phosphate, and let's put our stoichiometry up here. Okay, so I've got 3 of these. An epimerase, remember the difference that epimers differ in the configuration around only 1 carbon. So if you look at ribulose 5-phosphate and xylulose 5-phosphate, the only difference is carbon number 3, which has the OH pointing to the left. That's a very common theme in the pentose phosphate pathway. Here, we're going to convert a ketose to an aldose. So that's an isomerase reaction. So we start with 3 ribulose 5-phosphates, we get 2 xylulose 5-phosphates, and 1 ribose 5-phosphate. And again, notice that in the epimerase reaction, we've changed the configuration around carbon number 3. And for the isomerase reaction, we've converted a ketose to an aldose. So here are the reactions a little bit more up close, and here I've drawn this on ChemDraw to show that the only difference between the ribulose 5-phosphate and the xylulose 5-phosphate is that the configuration around carbon number 3. So the epimerase converts ribulose 5-phosphate to its epimer, xylulose 5-phosphate. And the isomerase converts the ketose to the aldose. So this is the summary of stage 2, or part of the non-oxidative branch. 3 ribulose 5-phosphates are what we start with. 2 of them are epimerized to xylulose 5-phosphate, and 1 is isomerized to ribulose 5-phosphate. So that's stage 2. Stage 3, what are we going to do in stage 3? Well, you might not have probably ribose 5-phosphate you've heard of, because we use that to make nucleotides. But what we're going to do is we're going to do some carbon shuffling reactions, and we're going to convert these compounds to fructose 6-phosphate and glyceraldehyde 3-phosphate. So hopefully those compounds look familiar. So we're going to do some carbon exchange reactions. And again, this is part of the non-oxidative branch, or stage 3. So we can summarize stage 3 in these reactions right here, in which we take 2 5-carbon sugars to form a 7-carbon sugar and a 3-carbon sugar. Then we take the 7-carbon sugar and the 3-carbon sugar to form a 6-carbon sugar and a 4-carbon sugar. We bring in a third 5-carbon sugar to react with the 4-carbon sugar, and we get a 6-carbon sugar and a 3-carbon sugar. If you add all of these up, what happens is you take 3 5-carbon sugars, that's 15 carbons, and you get 2 6-carbon sugars and 1 3-carbon sugar. So we're just shuffling carbons. So there's our 1, 2, 3. Those are our 3 5-carbon sugars that will be converted to 2 6-carbon sugars and a 3-carbon sugar. So this is the diagram in our book, and again, I'm going to show you just like this one piece at a time. I'm not sure that before we understand how this works that this is particularly illuminating, so we're going to come back to this. Okay, so the other thing that we should do, coming back to these reactions, is that there are enzymes involved. This reaction right here, the first reaction is catalyzed by a TransKetolase, the 7 by a Transaldolase, and then the third by a TransKetolase. So let's see how these enzymes work. Okay, so this reaction first. We're going to take 2 5-carbon sugars to give us a 7-carbon sugar and a 3-carbon sugar. What we should note in all of those 3 reactions that I showed you in the previous slide is that we're going to react a ketose plus an aldose as our reactants, and then we will get an aldose plus a ketose for our products. So what will happen is the aldose will form a ketose and the ketose will form an aldose. The other thing that we should note is in the first reaction, the enzyme is a TransKetolase, and this is what TransKetolase enzymes do. They transfer 2 carbons at a time, and the ketose is the donor and the aldose is the acceptor. So aldose and acceptor begin with A, so that's a nice way to remember that. And as we saw before, when you're drawing the structures, make sure that you draw the OH on the carbon number 3 and make sure it's pointing left in the Fischer configuration. Okay, so here we are with step 1. So we're going to take ribose 5-phosphate and xylulose 5-phosphate. Remember, those were 2 of the 3 products in stage 2. So we have 2 5-carbon sugars. We're going to use the enzyme a TransKetolase, and the TransKetolase transfers 2 carbons. So we're going to transfer 2 carbons from 1 5-carbon sugar to this next one. So we will be left with 3 carbons here and 7 carbons here. So we get glyceraldehyde 3-phosphate and acetoheptulose 7-phosphate. So this is how this works. So if we look up here, so up on top, I have the reactants. So, oh, I'm sorry, along the side, I'm very sorry. So on the left-hand side, I have the reactants. So our reactants are 2 5-carbon sugars. So here's the ketose drawn on top in green, and then this is the aldose drawn in black below. And it's a TransKetolase. I'm going to transfer 2 carbons at a time. And remember that TransKetolase transfers 2 carbons, and the ketose is the donor and the aldose is the acceptor. So that means I'm going to take these 2 carbons up on top right here. That's the ketose. And I'm going to give them to the aldose right here. So what happens is once these 2 carbons have been donated, I'm left with a 3-carbon sugar. And when this aldose accepts those 2 carbons, I have a 7-carbon sugar. And the ketose becomes an aldose, and the aldose becomes a ketose. And I know how to draw my structure of my ketose because the OH points to the left. So that's my glyceraldehyde 3-phosphate right here. That's my C3. And this is the acetoheptulose 7-phosphate. That's my C7. So I just showed you this first reaction right here, where the TransKetolase catalyzed the 2-carbon shuffling from one 5-carbon sugar to another, leaving us with a 7-carbon sugar and a 3-carbon sugar. Okay, what's next? Now we're going to take these 2 products, the 7-carbon sugar and the 3-carbon sugar, and we're going to use an aldolase this time to get a 6-carbon sugar and a 4-carbon sugar. So the enzyme is a transaldolase. It's the same as the TransKetolase except for one thing, and that is the transaldolase transfers 3 carbons at a time, not 2, but everything else is the same. The ketose is the donor, and the aldose is the acceptor, and the OH on the C3 of the ketose points left. So I have my 3-carbon sugar and my 7-carbon sugar. So here's the 3-carbon sugar and the 7-carbon sugar. So now what I'm going to do is I'm going to transfer 3 carbons because it's a transaldolase. So the 7-carbon sugar is going to donate, and then the 3-carbon sugar is going to accept, and now I'm left with 4 carbons over here and 6 carbons over here. So the 4-carbon sugar is erythrose 4-phosphate, and the 6-carbon sugar is fructose 6-phosphate. So I want you to have some practice on drawing these structures. So again, these are the reactants right here shown on the left. The aldose is shown in black and the ketose is shown in green. Remember the transaldolase, I abbreviated TA here, is going to transfer 3 carbons. And remember that the ketose is the donor and the aldose is the acceptor. So what you're going to want to do is you're going to want to draw the structure of the 4-carbon sugar left behind, and you're going to want to draw the structure of the 6-carbon sugar that you get when you donate these 3 carbons to this 3-carbon sugar. So you should stop the recording now and draw those structures. Well, hopefully you ended up with something like this, and I tried to keep my color coding. So again, these 3 carbons right here were transferred to this compound right here, and this should look familiar. This is fructose 6-phosphate. And the 4 carbons left behind, this is the erythrose 4-phosphate. And again, the glyceraldehyde 3-phosphate was an aldose, and now it's a ketose. And this acetoheptulose 7-phosphate was a ketose, and now it's an aldose. Okay, so that was this is the first reaction, that's the second reaction. Now we want to go to the third reaction. And what we want to do here is we're going to use this 4-carbon sugar right here. We're going to bring that over here. And this is our third 5-carbon sugar. Remember, we produced 3 5-carbon sugars in stage 2. And that's a TransKetolase. So we have erythrose 4-phosphate, that's the C4, and the xylulose 5-phosphate, that's the C5. And what's going to happen here is we're going to donate, remember this is a TransKetolase, we're going to donate 2 carbons this time. So the 5-carbon sugar donates 2 of its carbons to the 4-carbon sugar, and now we get a 6-carbon sugar. And we have a 3-carbon sugar left behind. So we're going to form a second fructose 6-phosphate, and we're going to form a glyceraldehyde 3-phosphate. So these are the reactants up here, and I've drawn the aldose over here in green and the ketose right here in black. So this is a TransKetolase, and the TransKetolase transfers 2 carbons at a time. The ketose is the donor and the aldose is the acceptor. So we're going to transfer 2 carbons from this this sugar to this one, and again, you should stop the recording here and draw the structures of the products. So hopefully you got something that looks like this. So if I have this 1, 2, 3, 4-carbon sugar right here, if it accepts 2 carbons from the ketose, we have something that looks like this. That again should look familiar. That's fructose 6-phosphate. And then these 3 carbons are left behind. That's glyceraldehyde 3-phosphate. And again, notice that the ketose became the aldose and the aldose became the ketose. So we are done. So in stage 3, we did those 3 reactions, a transketolase, a transaldolase, and transketolase. And what happened was we took 2 xylulose 5-phosphates and a ribose 5-phosphate, and our products were 2 fructose 6-phosphates and glyceraldehyde 3-phosphate. So to put this in some context now, in this figure right here, along the top right here, that's the oxidative branch. So those were the reactions involved in the oxidative branch. We started right here with the glucose 6-phosphate. So we did 1, 2, 3 reactions right here. The ring opening reaction isn't shown, but the 2 reactions that are shown are the 2 reactions that generated the NADPH. And remember, we oxidized 1 carbon fully to CO2 to get the ribulose 5-phosphate. And then we could take the ribulose 5-phosphate and in stage 2, we converted 3 of those, 2 of them to the xylulose 5-phosphate and 1 to ribose 5-phosphate. We can actually siphon off some of the ribose 5-phosphate for nucleotide biosynthesis. But in the stage 3 reactions that I just showed you, we have fructose 6-phosphate and glyceraldehyde 3-phosphate. And if you're going down, that's glycolysis, and if you're going up, that's gluconeogenesis, depending on the needs of the cell at the time. So this shows the connection between the pentose phosphate pathway and glycolysis and or gluconeogenesis. So I think this figure might make a little bit more sense now. So up at the top right here, that is the oxidative branch. So all 3 reactions are shown. The other thing that the artist does here is the artist draws 3 arrows to show that stoichiometrically we need 3 glucose 6-phosphates to go through here, so we get 3 ribulose 5-phosphates. Remember, we convert 1 of them to ribulose 5-phosphate and 2 of them to the xylulose 5-phosphate. So that's stage 2. And then this shows the transketolase, the transaldolase, and the transketolase. And then we end up with 2 fructose 6-phosphates and a glyceraldehyde 3-phosphate. And the 2 fructose 6-phosphate, one's over here and one's over here. So I think after going through the discussion we just did, I think this figure should probably make more sense now. So this is another figure that I like. So this shows the oxidative stage right here with glucose 6-phosphate being converted to ribulose 5-phosphate. Again, keep track of the stoichiometry because you need 3 of them. You'll get 1 ribose 5-phosphate and 2 xylulose 5-phosphates. And again, here we show the transketolase, the transaldolase, and the transketolase. And we end up with 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphate. So this is an interesting pathway in of itself, but what's more interesting, I think, is to think about how it's controlled and what the needs of the cell are, and what would the cell do if NADPH were needed and ribose were not needed. So what you want to do is you'd want to maximize your glucose to produce all of the NADPH that you could. So you should think about which pathways produce NADPH and not ribose. So this is what the cell does. You can go through stage 1, and if you go through stage 1, and this arrow right here, all 3 reactions aren't shown, but we convert glucose 6-phosphate to ribulose 5-phosphate. So there's our NADPH, and that's what's needed. Then we go through stage 2, where the ribulose 5-phosphate is converted to xylulose 5-phosphate and ribose 5-phosphate. We go to stage 3, those carbon shuffling reactions, and we get glyceraldehyde 3-phosphate and fructose 6-phosphate. And then we go back up, we borrow from gluconeogenesis, and we form glucose 6-phosphate again. And then we go through stage 1 again. And if we keep doing this cyclical type of pathway, eventually the net is we go through this stage 1 6 times. And every time you go through stage 1, you lose 1 carbon as CO2. So eventually every single carbon is completely oxidized to CO2. So if you added up all of those reactions and crossed out the intermediates, this is essentially what happens. So we can look at it this way, or we have 6 glucose 6-phosphates and 12 NADP pluses give us 5 glucose 6-phosphates and 12 NADPH. If we want to cross this out and just look at the net, what happens is 1 glucose 6-phosphate all 6 carbons are essentially completely oxidized to CO2. So that means you get 24 protons and 24 electrons. And the 24 protons, 12 of them end up here on the NADPH and 12 of them over here. And then all 24 electrons get donated to NADP plus to give us NADPH. So what we've done is we've completely oxidized every single carbon to CO2, and we've gotten the maximum amount of NADPH that we can from the glucose 6-phosphate. We don't get any ribose this way, but this is what the cell would do if NADPH were needed and ribose were not needed. So there is going to be some kind of partitioning between glycolysis and the pentose phosphate pathway. So I said that at the beginning. So if we have glucose 6-phosphate going through the pentose phosphate pathway, or to give us NADPH and pentose phosphates, or we can go through glycolysis and get ATP. So what happens is, if NADPH is forming faster than it's being used, either for biosynthetic reactions or glutathione reduction or reactive oxygen species or whatever, and the NADPH starts to rise, what happens is NADPH acts as an allosteric inhibitor of glucose 6-phosphate dehydrogenase. So if you block off this pathway right here, that means glucose 6-phosphate then gets shunted to glycolysis and you get ATP. So if NADPH is starting to pile up and you don't need it, then the glucose 6-phosphate is partitioned off to glycolysis instead. So that's one of the ways that the cell is able to regulate which way glucose 6-phosphate goes. Now, what if you needed ribose and you didn't need NADPH? Well, what you could do is you could carry out glycolysis first, and then get the, my arrow covers up the fructose 6-phosphate, but here's the glyceraldehyde 3-phosphate. So, and then what you do is you can go through stage 3 in reverse, because all of those reactions are reversible. So then you get xylulose 5-phosphate and ribose 5-phosphate, and then you can convert these pentose phosphates. Those are needed for nucleotide biosynthesis. So if you don't need NADPH, you wouldn't do stage 1 at all. You would just convert, you would use your glucose as a source of energy to form the fructose 6-phosphate and glyceraldehyde 3-phosphate intermediates that you would need to form the ribose 5-phosphate that is needed by the cell, probably for nucleotide biosynthesis. So that's what the glycolytic intermediates are used for. And if NADPH were not needed, then you wouldn't use the oxidative branch at all. So essentially what's happening here is you can take 5 glucose 6-phosphates, you need an ATP right here as an investment, and you get 6 ribose 5-phosphate. So essentially you're just shuffling carbons. You're taking 5 6-carbon sugars and converting them to 6 5-carbon sugars. So all of the available glucose 6-phosphates been isomerized to ribose 5-phosphate, and that's used for nucleic acid biosynthesis. And this would be if the cell needed ribose and didn't need NADPH. Well, what if you need NADPH and energy? Well, what you could do is you could do that partitioning, like I showed you before, where you can take glucose and send it through stage 1. That gives you the NADPH that you need. Then you go through stage 2 to convert the ribulose 5-phosphate into xylulose 5-phosphate and ribose 5-phosphate. Then go through stage 3 to get fructose 6-phosphate and glyceraldehyde 3-phosphate. And then you could continue through glycolysis, and that's going to give you some NADH and ATP. So this is what the cell would do. It would do if NADPH and energy are needed. So here the cell is essentially partitioning glucose through glycolysis and through the pentose phosphate pathway. So the distribution of the pentose phosphate pathway depends on the cell type. So the red blood cells do run this pathway because they need NADPH. It maintains glutathione in the reduced form. In red blood cells, you have lots of oxygen, so there's going to be lots and lots and lots of reactive oxygen species. So the glutathione is needed to convert those reactive oxygen species to something that's a little less toxic to the cell. Mammary gland, testis, adrenal cortex, they need NADPH for lipid and steroid biosynthesis. In the liver, that's where you have this balance between glycolysis and the pentose phosphate pathway, because the liver does quite a lot of biosynthesis and needs ATP and NADPH. The muscle does not have an active pentose phosphate pathway, so nearly all glucose 6-phosphate in the muscle goes to glycolysis to produce ATP to power the muscle, so very little to the pentose phosphate pathway. So as an exercise, you should draw the product of the following reaction. So this might be a little surprising, but here, if you follow the rules here, I have dihydroxyacetone phosphate, and this is a ketose, so it isn't CH2OH, there's a phosphate group here. And I drew it this way on purpose. And then this is erythrose 4-phosphate, that is an aldose. So remember, a transaldolase transfers 3 carbons at a time. The ketose is the donor and the aldose is the acceptor. So you should stop the recording and draw your product. And if you follow the rules, you get only one product. So what happens is all 3 of these carbons in this ketose, remember the ketose is the donor, get transferred to the aldose, and you form acetoheptylose 7-phosphate. And notice again that the OH on the carbon number 3 is pointing to the left. That is our whirlwind tour through the pentose phosphate pathway. I hope it was informative, and I will see you next time.